I'm going to tell you just in a necessarily abbreviated fashion today the story of a curious observation that turned into an obsession, which turned into a worldwide resource for HIV research. And the database is currently PI'd by these lovely people with the support of staff, and it's located at Los Alamos National Laboratory, which is the birthplace of the bomb, and we are sort of a pimple on the behind of the nuclear industrial complex. But there are a lot of nice things actually about being located at a national laboratory, which I will talk to you about over a beer sometime. So the database, as I said, is a long-standing project. It gets a lot of use, tens of thousands of uses, visits per year from researchers on essentially with a worldwide distribution. And we have, of course, a large number of HIV sequences in the database. We're pushing 1 million. We're planning a party for when we cross that threshold. 1 million sequences, also with a worldwide distribution. No samples from Antarctica yet, but we've got all the other continents. So the key point here, and I'm going to just be stressing this over and over again, is this is an integrated effort. So we present both the HIV sequence data from the pathogen and the immunological data from the host. And because the essential feature of this epidemic is the coevolution of the virus and the host immune response. And so for any sort of vaccinological studies, for instance, it's critically important to understand that evolution, as well as the epidemiological effects of viral diversity and human HLA diversity. So in addition to the sequence database, we have the Immunology Database, which records responses not just to HIV, but and not just in humans, but also responses to other primate immunodeficiency viruses. I mean, I feel like Hobbes the tiger, who, well, can you draw anything besides tigers? Oh, sure, he says. Pumas, ocelots, leopards, you name it, right? We do any primate immunodeficiency virus, with a huge diversity of that. But as I said, large number of epitopes, both CD8, CD4, a bunch of information about monoclonal and other antibodies, and a selection of tools that are basically a large set of tools, most of which have arisen as a research question, either in-house or by one of our database users. Said, oh, wouldn't it be nice to do this? And oh, sure, we say we can figure out how to do that analysis. And once we've figured out how to do it, then we can essentially build an interface so that you just push a button and you can do that. Of course, our big thing is first you have to build the button. Right? Once we build the button, anybody can push it. And these tools are a mix, depending on how you slice it. This is probably wrong, between tools that are actually specific for HIV or other primate lentiviruses, and tools that are really available for any pathogen or broadly speaking, any immune response. So so here's where we are today. Today I'm going to be giving a sort of 30,000-foot overview, and then my colleague Elizabeth Sharon will be giving a talk on essentially how the Tcell epitope data is stored and entered, and how to search it, and some of the peptide tools that are most relevant to CTL responses. Tomorrow I'll be talking a lot about antibody-related data and resources, and some of the other tools, and we'll get to a little bit of vaccine design, which is my primary interest and the interests of a number of people here. So today, when you go to our homepage, and that's it, and you can all do it. You don't have to, but you can. You have a choice. So we have several. The two major databases: the sequence database, Immunology Database. You can click on one or the other, but the key point is they are tightly integrated. So I alluded to this earlier. Jerry Myers in the theoretical biology group at Los Alamos noticed in 1983 the first sequence of HIV envelope became available, and he thought, oh well, that's interesting. And at that point, GenBank, which was started in this group, was present there, and so he looked at that sequence, thought, oh, that's interesting. Then the second one came out shortly thereafter, and it was different. And when the third one came out, and it was different again, Jerry said, well, we may have a problem here. And in fact, so he started collecting these sequences as they came out and collating them. And it got bigger and bigger and eventually became a project with separate funding. Betty Korber, who started as a postdoc with Jerry in, I think, 1990, took over the project in 94 and founded the Immunology Database to govern the corresponding diversity of immune responses in 94, and subsequently took over the sequence project as well. Now, so as I said, I'm going to be giving, for all of this, we're going to be giving a sort of abbreviated version of the rest of the day's talks. So the IDB, our IDB colleagues have been going through in some detail what's what's in the database and really how you use it. We're going to be much briefer than that, and I'm just going to try and give you the 30,000 view of what's in there, with occasional dives down towards the surface so you can take a quick look out the window. And these are some of the tools we'll be referring to tomorrow, primarily tomorrow. The key point is, I'm not going over all these slides. Elizabeth's not going all over all these slides in detail. A lot of the stuff is available for you to look at later at your leisure to see things that oh, that might be interesting for us to use. So I'm going to talk about two applications here. So the key point is that although neglecting the search, about a fifth of our tools are specific to HIV. If you don't care about HIV, those are not going to be relevant to you. But the remaining 4/5 are either partially or fully applicable to any virus, or they're strictly immunologically directed tools. I'm going to point out that a tool, some methods we developed for specifically for HIV, to develop vaccines for HIV, have been, and I'll talk about them a little bit tomorrow, have been extended to other pathogens. So these mosaic or epigraph vaccines have been used to make rabies vaccines, filovirus vaccines, vaccines for the bacterial pathogen chlamydia. They're being used in pigs to prevent this this terrible, unpronounceable disease. And these are both our group, vaccines designed by our group, and vaccines designed by other groups. And I'm going to brag a little bit. I have to, because this makes me so happy that our HIV mosaic design is now moving into phase 3 human trials, which is cool, because that means it may. We sent this design out in 2007, and now 12 years later, we're about to find out within the next year or two whether it works. The other thing to point out is that we've got a database structure and tools for sequence analysis and immunology and the comparison, which are portable in the computer science sense. That is, they can be ported to other viruses, and we have done this to varying degrees. First with Hep C, later with a set of hemorrhagic fever viruses broadly defined. After the 2014 Ebola outbreak in West Africa, we got a little bit of extra funding to update the filovirus sequence database, which is again something that has been useful to people working on that heinous virus worldwide. Currently, the only one that's under active development is the HIV database, but the sequence portions of the other databases are being continuously updated. I'll mention as an high level view, our operation is similar to the IEDB operation in that we rely on public data and add value to it. So we're taking immunology data from PubMed, and our annotators are processing that and extracting information and putting it into the the context of HIV specifically, and we're harvesting sequence data from GenBank, and again adding value to it by properly subtyping and putting in context and putting gene maps and a number of other detecting recombinants, doing quality control, a number of other important tasks. So I'm going to take you very quickly through the different categories of tools we have. And point here is that they're color-coded by what they're good for. So if it's red, it's pretty much only HIV or HIV and SIV. Purple, it's it's got an HIV bias, but still likely to be useful. And blue, it's pure general use tool. You can use it for anything. So the analysis and quality control tools essentially allow you to quantify a number of important metrics, mostly about sequence data, but a lot of immunologically relevant things. For example, N-link glycosylation sites, which are huge in terms of antibody development, sequence variation, pattern analysis, a variety of entropy, I'll point out, a variety of evolutionarily important metrics. Phylogenetics, again, is a long-standing interest of our group, and we have a number of tools for that. I'll just point out that if you have a sequence-based study involving any pathogen, you should be doing some phylogenetics. Sometimes people omit this, and it gets them into trouble. And these are, again, mostly all general use, except for this guy, which basically tells you what you have and what your HIV sequence is. The others are quite general. Format and display, these are essentially convenience functions to deal with some of the more maddening aspects of bioinformatics. File formats, oh my goodness, and a number of other important and useful things that are difficult to code up yourself. I'll point out gene cutter and sequence locator for the HIV people, critical, but a lot of alignment manipulation, again, very important, and a pain. Finally, what's the most interest to this particular talk are the database search interfaces and the immunology tools. So we'll spend most of our time on this particular area. And again, I'll just be dipping down and looking at some of these in more detail. So, as I said, we harvest data from the public literature, and we break it up in this sort of obvious way. There are two arms to the adaptive immune system, and so we have two classes of data. We've got the Tcell, both CTL, CD8 positive and helpers, CD4 positive epitopes, and they're organized in slightly different ways. The CTL epitopes are organized essentially with one paper gets one entry. So one person has reported this epitope in this person or this group of people. That's one epitope, one entry, and we'll get if you go and search for that epitope, you'll get a number of different studies for that particular epitope. Whereas the B cells, because there's a lot of work with monoclonals, we get essentially one entry per monoclonal antibody, and all the studies referencing that antibody get linked to that single entry. And again, I won't dwell on this too much, but this is similar to the approach that IDB has taken, though they're a little more formal in some of their, in the use of ontologies, for instance. But we store the essential information, see what it, what it is, where it came from, and how it was delivered, and variants, who's recognizing it and how. And we go into, I'm going to go into some detail tomorrow about the neutralizing antibody resources, particularly CatNap, which shows things like contact residues, what are the neutralization, what sites are important for neutralization, and those may or may not be contact sites. And again, there are everything that doesn't fit neatly into an existing field is recorded on a summary note, which is not not the abstract of the paper, but the relevant facts from based on the judgment of the annotator. And there are one of the classic things in Unix. Are there any sort of Unix identifying people here? Is it there's more than one way to do it? Thank you. Good. Got at least one. And so we've analogously to that philosophy, there are multiple ways in to both ways to navigate the database and ways to approach different approach the same question. You can go multiple ways, different search interfaces, and a variety of tools, some of which overlap with different emphases. So I'm going to let Elizabeth go over these things today, and I'll be going over much of this tomorrow. And with that, I think I will turn it over to my colleague. So this is again your entry page to the database, and where you have several choices, but none of them are irrevocable. So my colleague Elizabeth Sharon Fung is a PhD immunologist, annotator at the database, and is responsible for entering a lot of the data that you will see later in the day. So. Now that I'm safely off stage, feel free to interrupt the questions at any time.
We'll call him back.
And to be fair, we'll also have breakout time later at the breakout sessions to answer individual questions and go through stuff with you. Thank you.
Okay, so Will showed you how we have a lot of data in the HIV database, and I'm here to show you just briefly how you can search for that or how you can access some of that data. So here is the page that you would be taken to if you type in hiv.lanl.gov. And so that's given down at the bottom, and if you choose to, you can follow through and see this. And you can see immediately that one of the strengths of this database is that there's a lot of crosstalk between the sequence and the immunology database. This is so important in HIV, where the sequences are just blossoming and blooming, and because of that, the immunology changes as well. And this is the HIV sequences. And then we'll already talk to you about how you can access other databases like HCV and the filovirus sequence databases and some of that immunology. So one of the things that we have as well is that if an epitope or an immunological site is annotated with just the sequence numbers and not any real protein sequence, we can actually put it in because we have a reference sequence, and it's called the HXB2. And so you'll see that later on again and again, that any immunological HIV sequence can be aligned to this reference sequence that was a first isolate and then put into the database. And then, like IDB, we also try to follow through and torture authors and ask them to send us more information. So if you were interested in, let's say, the immunology, which is what I'm here to talk about, if you click on that, then you get onto a page which has all these different abilities and things that you can do. You'll see immediately that some of the things we can do is you can get a summary of all the data, depending on whether you're interested in CTL or helper T epitopes, or whether you're interested in antibody information. and you can get the whole database information summarized, or you can do your own searches, which is of course what most people like to do. We have, in addition to that, some different sets of data, and you can also go to different tools. And these are just PDFs or links to other people's studies that are very important, and that you can actually go in and get all their data, because they've chosen to very kindly allow us to have their data up here. And this is a very great resource, actually. Another thing is that under HLA-TEM, we have several sets of data from different groups which have data about HLA polymorphisms and HIV. So these are the different tools and things you can get. Well, so you see that you can actually search, or you can look at all the summaries of the data we have. So if you're looking at summaries of data, let's just say you want to look at everything there is in the database, there's a diagrammatic way to look at it, and there's also a tabular form. And the diagrammatic way, the best way, is to look at maps. And then the tabular form is epitope tables. And here I'm talking about T-cell epitopes. I'm not yet into antibody. We also have antibody tables, and there's a few antibody epitopes. You know, as time goes on, there's more and more. Okay. The next slide I have here is different ways, as Will pointed out, there are different ways of maneuvering and going through to get the same thing. And so you can use a drop-down. Whoops! What is that? Sorry. There we go. You can use a drop-down menu at the top if you want to, say, look at different products. Or you can also just click to a link. So there's very many ways of getting to the same place. Just choose your favorite way. And if you were to choose, say, tables, which is one way of summarizing data, then you could look at either CTL tables, you could look at helper T-cell tables of epitopes. You could also look at antibody epitopes. And then we have a different table, which is very interesting, and that is the variant epitope table. And this is because it's very important, because in HIV, the epitopes mutate so very quickly. And so we have tables which are updated by us frequently, where epitope variants are present. And so we have helper variants, and we also have CTL variants. Okay, let's see if I've got everything here. Here is another way of summarizing the data, and this is on epitope maps. And here I'm talking about T-cell epitopes. And this is of a CTL map and this is, we have it in the database given according to the different HIV proteins. And here's just a little screenshot of a map. You can see that it's very, very complex. There's just tons of data. And what you can see also is that we've marked on the reference sequence here where exactly these epitopes lie in reference to that epitope sequence. So here you don't actually see the sequence of the epitope, and the epitope being this 2-arrow bounded line, which is about 14 amino acids or less or more, usually 14 amino acids and less. And you also have the restricting HLA above it. And so you have the HLA, you have the bounding of the sequence, and the neat thing is it's clickable. So I can't do it here, but if you were actually at this map and you were to click it, you could go then to information about that particular epitope. And from there, if you wanted to, one click away is information on how that epitope aligns to pretty much all the sequences in our database that have this particular epitope. And these epitopes would align also to sequences that include variants. So a small change in an amino acid or something like that. You start out with identity and then go down from it. So it's pretty powerful, where you can go from the epitope that you searched on or something like that, and then you just go on into clicking your way into where exactly this epitope falls in the genome. Okay. So interactive maps are a big boon. And let's go on to tables again. If you were looking, and this is only true for CTL, so Tcell, cytotoxic epitopes, we have two different tabular summaries of the data of all the epitopes. One is just a very comprehensive table, and we call it the B list, and it has pretty much every epitope that this is only a small snapshot, this is not everything, but it has every epitope that we've curated within the database, and it has information about it, like what protein is it from, what's the location in reference to our HXV2 sequence, what subtype, species, and what HLA restricts that particular epitope if it's known. The other epitope table that's very, very useful is called the A list, and this one is a very refined table with very, very defined epitopes. And the two major defining things for this table are, one, these epitopes have been studied through truncation. So they are the most potent form of that particular epitope. And then the second thing is that they have been experimentally verified with HLA restriction. So these are what we would call, if you're an experimentalist, your gold standard epitopes. We know this is right, and this is curated and kept by Christian Brander and colleagues. And almost every year we have an update. He puts out a review, which you can find on our website. And then this table, this A-list table, is updated, and it has a lot of epitopes now in it. But if you're looking for just everything, then the B-list table is where you would go if you were looking for the summary of all the data. The next thing you can do is you can search. So we have summaries of our whole database information, the Immunology Database information. You can find that on maps or in tables. And then if you want, you can do a search. This is what our search form looks like. And you can see that you can search on many things. You can search on the protein of interest, or you can search on the particular epitope you're actually interested in. You can just search on a location, names of the epitope, and you can also search on different subtypes. You can search on the immunogen. And then because there's so much of the database is devoted to vaccine predictions and vaccine development, there are different vaccine details that you can search on. Species would just be humanized mouse or human, or where was the study that this epitope came from, in what species was it done. And then there's HLA, author, first author or last author. And if you're interested in just anybody who might have done the work, just don't check any boxes, and you can search on author name. There's different countries, and then keywords and notes are there. So under notes, what you would put is just a word that you consider a keyword for your study. And then if that was reflected in any of our notes, then you would be able to find all those records that correlate with epitopes that have that word in its notes. Under keywords, these are keywords that are not the standard keywords that you find under a paper when you pull it up from PubMed. These are actually keywords that when we read the papers, we determine that these are keywords in relation to the study of that particular epitope. So sometimes you will find, it may be an early acute infection, and that may not be a keyword that the author actually flagged. But if we find it to be a keyword, we do put it down, and then you can actually search on that. So that actually broadens the search for you in your particular interest. Could also make it more specific. Okay, you can also double dip when you search, and by that I mean if you were to, there's a very famous HIV epitope, SL9, this one, Slint-Vattel, and if you put that in and searched on it a number of years ago, you got 285 entries. Now we have even more. But if you did not want to see every single SL9 result, then you could put in SL9, and then you could put in, say, escape, because you're only interested in where it escaped. And so you put that in, you get only 35 entries, and that's a little easier to deal with than before. So if you want to follow in and see how a search is done, I'd say search for this word, this epitope, IW9. And if you put that in here, it's ISPRTLNAW. And then if you restricted it by putting in the first author Pille, then you could see what you would get in the next slide. And there's one thing I forgot to mention, and that is wherever there is a URL, we have it in pink here, and that would be the link that you would want to get to this particular search page or whatever it's on. Okay, so this is the result that would come. Now, because the search was restricted from IW9 to just that first author, Pille, we just get one record. As Will had said earlier, all our different epitope records are singular. So each epitope in a different study is given one record number. However, that's not the case with antibodies because of the nature of how the data comes. With antibodies, every antibody is its own record, and then each study is a different note. So you could have one antibody with 100 notes, but you could have one epitope with 100 different records. So it's not as confusing as in some ways. It's just our definitions of it and how we organize it, but when you click through, it'll all make sense to you. So this is the resulting record that would be pulled up. This is really where the meat of your search results resides. And you could see here that, it says at the top how many matching records there are. You could have several. In this case, it's just one. It gives you a record number, which really is a reference for you to pull it up out of the database whenever you want. It gives you a nice location moniker there where, this is the location where in alignment to our HXB2 reference sequence, but this is the location that the author coded. And most often there's 100% agreement between the two, but sometimes there is not. Here's the epitope. It gives you the subtype, the species, what the immunogen was. It was from an infection. And here's something important. This is the donor HLA. And what that really means is it's the subject of the patient's HLA. We call it donor. It means it was the subject from which this particular epitope with its response was found. And so all that person's HLA are given. There is another category which is just HLA, and that is the restricting HLA. But this is all the different HLA of that particular subject. I know someone else had asked earlier today about that. And then we say which country it's from, and it's not shown here, but we also have studies. So this one doesn't come from a particular study, but there are studies, very large HIV studies that are done, and that is also recorded. And if it is there, it will pull it up. For example, was it a Chavi study or a Caprisa study? Was it a Botswana cohort? Where did this come from? If we have it, we annotate that. And then we have keywords. And you can see here this was a response in a child. There was mother-to-infant transmission. There was escape. And it was an acute early infection. So many, many keywords that you could possibly pull up a record with. And then we also have notes. These notes are, and I know Will said this, they're not the abstract. They include important things from the abstract, but we try to have the first note. I did it again. I think I was here, wasn't I? Yes. We try to have the note be just something that gives a summary of the paper, and then it talks about that particular epitope and what that particular epitope's information and things are. And so you can see that there's much more that we pull out. And these notes come from figures, from tables, from supplements, and of course the body of the paper as well. And then at the bottom you find references. The neat thing about the references is you can go straight to the PubMed from there, from clicking on the PubMed ID, or you can see all the other entries that particular paper had. And this becomes very interesting when there are other epitopes that were mentioned, sometimes overlapping, sometimes not. Okay. Well, you can see here also that on the right of this page, there are certain links that you can go to. You can go to the epitope map, where you will see where this particular epitope resides on the map. And this is similar to the picture I'd shown way earlier with the clickable epitopes. Or you can go to alignments or variants. And this is if there is a variant for this particular epitope. And by variant, I mean that there is at least one amino acid difference from the canonical or from the annotated epitope here. And not just that, sometimes these variants are escapes. Okay, let's go on to the next one. So I'm going to show you first about variants and then about epitope alignments. So you can actually get to variants by going to a certain epitope and then clicking on its variant information, kind of like how we did right now. Or you can go straight to the variant table and go find variants there. Here is a record with variants, and you can see that we have a note there too. This note will be very specific for that variant of the epitope, and that has been annotated. You can see how that variant was found, and something very key here is that we give it a mutation type. And this is because mutations can be a reversion, you can have a deletion, you could have an escape, and these variant mutation types, we give it this name based on what the author says. So if the author says it's an escape, we'll call it an escape, even if we, from reading the paper, don't quite agree with that. There's a lot of fudginess over here because it's kind of a definition you give it, and so for example, I may look at something and think, oh, that's a diminished response to that epitope, but the author may swear it's a bona fide escape. And so we just give what the author said over here, but in the note, we would mention, author calls this an epitope, but we see a diminished response in the tables of results. So that's that for the variant, different searches on variants. And different kinds of mutation types are like escape, inferred escape, susceptible form, and there's a ton more. Also, the way we denote a variant is that we have a lowercase letter where there's a change in or a mutation. Okay. And let's now go to alignments. So I'm going to go back actually, and you can see here when you come to a record of an epitope, it says epitope alignment. This is what it would look like if you clicked onto it. Now, this is not of that same epitope, it's a different one. This is SL9s. And you get to the output of a tool we have called Quick Align. And here you can see alignments in many different forms. You can see it as a web logo with different positions and what the probability of seeing a certain amino acid at that position is. You can see summaries for alignments to that epitope. You can see tables, and you can even see just a slice of the alignment and follow through on that. There is also another alignment tool called Analyze Align, which is newer, has more associated features with it, and that one can also be used when you use that particular tool. You use it in coordination with giving it different, coordinates. So you actually give sequence coordinates, and that's how you get alignments from it. Okay? All right, so Analyze Align is more, you can do a lot more with Analyze Align than you could with Quick Align. Both of them can deal with discontinuous epitopes, which is very important, especially when you're thinking of antibodies. Now I just want to show you a few tools that we have, just a couple. I'm not going to show you much at all, but these can be used on sequences and things. One of them is called PEP Gen. This is a really cool tool, and I know that anyone in an experimental lab would love this. And that is that if you give in sequences that align, you can then ask for overlapping peptides to be generated. You can say how long you want them to be. You can say how long you want the overlap to be. And you can ask for duplicates to be removed. And so for large studies, this is really important and useful. Another tool that we have is called Epitope Location Finder. And what this one does is it helps you, if you put in a sequence that you're interested in, it will help you based on HLA motifs. It will help you to find predicted epitopes. And this is for T cells. So when you do the search, you can actually even do an IEDB search, and thank you to IDB for this. So this is how it links in with our database, and you can check to use the IEDB search, or you can just check to use ours, or do both. And in addition to this, we also have something related called motif scan, where you can actually do a search on motifs that are HLA restricted motifs based on HLA restriction, and then it aligns it to a reference sequence that we have. Otherwise, you can just give your own sequences. And the result of this would give you something like this, where the epitopes at the top are actually completely matching epitopes. And then you have successive ones that are less and less of a match. And it tells you how much. If you click align, you go to the alignment of that epitope against all the other sequences that you've provided or that we have, if it's HIV. And then finally, you can go further, and this is, if you had checked the IEDB box, this is the result you would get from that search, and this takes you directly to IDB's website. And this will look more familiar to you because it's the IDB format with the table, and it gives you the percentile rank of that particular epitope and its prediction. Okay? So thank you for listening, and we just wanted to end on this to say we are so fortunate to be able to use this link with IDB and use it for our epitopes and things. And these are some of the things we have. If you have any questions, or if you have anything else you would like to find out about how to use our database, please let us know. And also let us know what else you would like to hear about from our database. I think we have a couple minutes if there are any questions for either Will or me. Yes. Go ahead.
Two questions. Number one, you spoke about the Tcell epitopes.
Mm-hmm.
Is that information linked to some groups or the individual's laboratory where the isolates can be tracked?
So what you can do is, on the sequence database, that information is available. It's always linked to the publishing group. So you can do that. And on the immunology database, at the bottom of each record is mentioned the paper that it came from, and from that you can find...
That I understand, but would there be a database for collection of different isolates in HIV field? I don't work on HIV.
Yes, the sequence databases.
So is that linked to this, your toolbox?
Yes, if there's a sequence, if there are sequences, there will be a little clickable link that you can go to, and it'll take you to the sequence database.
Okay.
Yeah.
Second question, sorry.
Will may have more to say about that, if you do.
No, I think you covered that correctly.
That's it. Okay.
Okay. Second small question.
Mm-hmm.
You spoke about the linkage between IEDB and your database. Except that, would there be any overlaps, to save some time? Meaning to look for the information.
So I believe their prediction tool is more sophisticated than ours, and so you could go to theirs. Will can tell you more about this, but this is only as far as epitope prediction here. We have many other tools that there's absolutely no overlap with. So, but in this one, yes, I don't think there would be an overlap except maybe for the first few that you pull up, the predicted epitopes. And Will, did you have something to say about that?
Well, our prediction tools, such as they are primarily just motif-based. They're not, as Elizabeth said, very sophisticated. So we're happy to link to the IDB tools for that.